Okay, thank you very much, and thank you to Cameron and to Marianne for inviting me to speak today. I'm going to talk very briefly about and give an overview of some of the work that we've been doing over the past 18 months, which is modelling diagnostics for global health. As many of you will be aware, particularly those who are in the drugs and the vaccine development fields, mathematical modelling, whether it be PK/PD modelling for drugs, or even the sort of public health impact modelling that I'm involved in, it's increasingly being used not just to say where are things going in the field, but also to go backwards and think about target product profiles and what are the new tools that need to be developed and what sort of profile would they have and where would they be useful. So in some sense it's a market analysis. The key feature in infectious diseases really is that interventions have both a direct and indirect effect, and that's why the models tend to be a little bit more complex than the standard other models. And cost-effectiveness outcomes can be added to these to look and rank prioritise different products, either for development and then also for implementation. And much of that sort of work informs the guidance produced by people like NICE in the UK or somewhere like Gavi internationally. So this has become a very standard thing in the field of treatment, drugs and vaccines, but there really hasn't been an equivalent methodology in the diagnostics field. And in particular, and I think the NTD community are very, very aware of this, even with the limited diagnostics there are, it's increasingly the case that diagnostic is no longer just used at point of care in clinical practice to say, has somebody or have they not got this particular infection, but is being used either to monitor and progress elimination in the field or to target different interventions. And so the product profile that one might need in that sense could be quite different. So we ran a project with the Gates Foundation, funded by them, over the last 18 months, it's now just about closed, really to try and think about and stimulate this across a number of disease areas. So it had two aims, really, to support the development of these sort of models for decision-making for product development, very much focused at the time on the Foundation's portfolio, but then also to get the different disease areas together, because diseases tend to end up in vertical programs and not cross-share the different ideas. So we were pulling different groups together from a range of infectious diseases. And I'm not going to talk about all of them. We did look at diagnostics for HIV, for malaria, for visceral leishmaniasis, that I hope maybe Emily will mention later, and prognostics for pneumonia. And all of this work will be coming together and should be published in a Nature supplement towards the end of the year, with some additional work on Ebola, in particular about response times, and also tuberculosis. So I'm going to really focus on malaria because that's my area, but I think quite a lot of what we're talking about in malaria really does map onto a lot of the NTDs, because there is a renewed focus on malaria using mass drug administration or mass screen and treat programs in the field for malaria to move towards elimination. So this work, it's a wide group of work, and this particular piece of work was in collaboration with school colleagues at the London School, was first of all to say what is the current state of diagnostics in their field, and in particular, not how well do they diagnose someone who's sick, but how well are they detecting those who are asymptomatic and potentially are going to go on and infect others. So a lot of previous work had been done by my colleague Lucy O'Kell in this area, looking at the relationship between standard light microscopy, that was really the old standard in malaria, and PCR, various types of PCR, but all much higher sensitivity and lab-based. So this is the curve that she found with PCR prevalence on the bottom and slide prevalence along the y-axis. And in general, she found that only around 50% of infections that were present by PCR would be detected by light microscopy. So that already gave us a fairly large bunch of infections that are going undetected. Now, microscopy is still used in many places, but often rapid diagnostic tests, which are antigen-based, are now being used in the field. So the first step was really to update this to look at RDTs. And what we found, the graph on the left shows, again, the microscopy prevalence on the x-axis and then the RDT prevalence on the y-axis. This is from a very large sample, majority children under 5 across Africa, because this is now done at a population level in many of the DHS surveys. So you can see the dotted line there is showing the sort of line of equality. So you can see pretty much RDTs have a sensitivity, it's a very slightly higher, but not dissimilar to microscopy. And obviously then, that, by comparison, which you see on the next slide, that's somewhat lower than a PCR. So still RDTs will be missing a substantial pool of infections. Interestingly, the RDT sensitivity was better in children than adults. That's perhaps not that surprising. Children tend to have higher parasite densities than adults, and that's what's shown on the right. So then we pulled those two things together and said, is this all consistent? If we've compared microscopy and PCR, and we've compared RDTs and PCR, does this all fit together? And actually, they all triangulated quite well. So this is showing the different relationships. The dark blue dots, which I think you can see towards the bottom, the dark blue line is the RDT PCR relationship directly from the observed. The pink line is the RDT PCR relationship if we went through microscopy and tried to predict it. So giving the same information, but again, dramatically showing you that you're probably missing quite a few infections in the population using either an RDT or microscopy. And this just shows it again at different transmission levels. So malaria is quite heterogeneous. There's a variation in intensity of transmission in different parts of the world. So at very low prevalence, we do seem to be missing quite a lot of infection. So everything below this line is our sort of iceberg, the tip of the iceberg, which is a common picture used now for this. And you can see it's even higher at the very high prevalence on the right-hand side, and particularly in adults. So there is a real pool of infection out there that if we're going to use strategies that are aiming to clear the parasite reservoir, we'll need a new diagnostic tool. So there's the infections. So then we wanted to go on and think about what is the product profile for this new diagnostic? How sensitive would it really need to be to be useful in the field? And the first step to doing this is to think about who's going to contribute onwardly in terms of infection. So I've showed you who's infected, and we need to translate the density of those infections on to how many mosquitoes they go on and infect. So this was a data set we used from a study led by Tern Bousema, looking in Burkina Faso at a cross-section of the population. And we stratified them in the left-hand column into whether they had disease, so very high parasite density. They were asymptomatic but detectable on an RDT or microscopy. And then the bottom is asymptomatic, undetectable by the RDT or microscopy, but still infected by PCR. The second graph shows you the gametocyte densities. That's the onward infectious stage for falciparum. And you can see these correlate, but there's quite a lot of variation there. And in fact, we found from a range of studies that parasite density rather than gametocyte density seems to be a better predictor of onward infectivity. So if you go to the fourth column, so one from the right, this then is the proportion of mosquitoes. These were all fed on the infected blood. And the proportion of mosquitoes infected by the individuals in that group. And you can see that still the clinical disease with the highest parasite density is contributing a high amount in terms of mosquito infection, so 27% infected mosquitoes, and it gets lower as the parasite density gets lower. But then you need to multiply the size of the group, how many people have clinical disease or asymptomatic infection at any point in time, to get to the right-hand column, which is the contribution of that group to onward infectivity, and that's actually the number that determines the reproduction number that we're wanting to drive below 1. And so that really shows that the dominant group is the asymptomatics that are detectable at the moment, and so therefore in some centres the RDTs will be getting a large part of that, so in total around 70% in this setting. But the asymptomatic undetectable is still a very large pool, and we need to find some sort of tool to be able to identify those individuals. So then we went on to think about looking at that in a slightly different way, and think about how we would then shift the curve down and have lower sensitivity tests and what they would detect. So the graph on the left-hand side is showing that same data, but is now showing the distribution rather than just averages. So you can see the parasite densities on the x-axis are very high for clinical disease. That's the big red spike. They're lower but quite variable, so that was partly variation in detectability. And then a pool of very low density infections. And you can translate that into a sort of Kaplan-Meier plot as you go up. If you go from the right-hand side of the middle plot, that's the diagnostic threshold sensitivity. As you go up, you start as you increase your sensitivity, go down in terms of that level, you start detecting more infections. And the interest then is to work out how many of those you'll detect at a given threshold. So if we took a detection limit, say, of 20 parasites per microliter, that would detect all of those individuals. And you can see that on the curve there. You can see that that would detect all the clinical disease. It would detect quite a fair proportion of those that are currently detectable in using microscopy RDT, a large proportion of those, and would detect around 50% of that undetectable, low-level infections. And the graph on the right showing you the proportion of the total onwards infectivity detected. And similarly, we can do that for different thresholds and get an estimate of how many infections a new product, a higher sensitivity test, would detect. So based on parasite densities, the summary of that was that if we increased it by 1 log order, so to 20 parasites per microliter, we'd get an increase in the infectious reservoir detected, at least in that setting, from around 50 to 90%. But increasing it as another log order would get us up to around 95% of the infectious reservoir, which is quite a large portion. Then the interesting thing is those curves, those Kaplan-Meier curves, flattened out. So you'd have to put an awful lot of effort in, get a huge sensitivity increase to buy that last 5%. So probably the cutoff point is around the 2 log order difference. And then finally, we wanted then to relate that to what impact this could have in an elimination setting. And there are 2 different ways that these mass drug administration scenarios are being trialled at the moment, and this is ongoing in Zambia. It's either through a mass drug administration where you don't care about the diagnostic, and that tends to, of course, always to be more effective, more people would be treated. But we're interested also in a mass screen and treat, because being repeatedly treated isn't always acceptable to the communities. And so the graph is showing you the places where you could interrupt transmission. So on the bottom axis is annual entomological inoculation rate. That's a measure of intensity of transmission. And the y-axis is the number of rounds, how many years you need to repeat this program until you achieve elimination. So you can see for the MDA, which is the purple line, if you read across that, at an annual EIR of around 2, you'd only need 2 rounds. At a slightly higher transmission, you'd need maybe 3 rounds and so on before you could interrupt transmission. The green line is showing the current RDTs if used in a mass screen and treat. And you can see there that it's really quite difficult. Even at an EIR of 1, you're going to need at least 8 rounds, so 8 years of keeping this program up at high coverage. And these all had quite high operational coverage assumptions to them. And bringing it down to the red line is going to both increase the speed at which one could reach elimination and also increase the geographic range at which this strategy might be a useful addition. Okay, so just to conclude, this gave you an example, but I think a lot of the same sort of work can be done in the wider NTD fields, thinking about what the target product profile could be and how that then relates to the public health benefit. And it is a tool that really is used in as I said at the beginning, in the drugs and vaccines field, and I think the diagnostics field has lacked a little bit of this quantitative input. So just to summarise, for the malaria side of things, we know that RDTs have a similar but slightly higher sensitivity than microscopy, but both are missing these infections. And even just a 1 log order increase in the sensitivity of these RDTs could substantially help to both expand the geographic regions and decrease the time to elimination. Finally, I'd like to thank the very wide number of contributors. I've just put here the people that worked on this particular malaria example, but we had groups across all of these infectious diseases, I think about 50 or 60 people in total. But in particular, the colleagues, both in Imperial and at London School, who did a lot of the work comparing the RDTs and PCR. To the two other modelling groups at Mahidol and Swiss TPH, who provided comparative results, so we did actually compare across models the data that was collected by Andre Lin and Robert Sauerwein, and a lot of input from the product development side from Paul Labare at PATH, and finally funding from the Gates Foundation. Thank you.